Hello and welcome to our lecture on viruses. Today we will be talking about fun things like rabies and the flu and Ebola. But before we can do that, we need to first talk about what is a virus. So if we break it down to its basic form, a virus is basically an infectious piece of genetic material inside a protein coat. Alright, so if that's what it is, what does this look like? If you haven't done so yet, go ahead and pause this video and paste in your virus diagram into your notebook. Once you've done that, we will go ahead and label it. So we're going to actually start from the outside of this virus and work in. So on the very outside, these things sticking off of it are the glycoprotein spikes. And if we break this word down, protein obviously means protein. And we see glyco or gluco in reference to sugar. So these are spikes that are made of carbohydrates and sugar. And what these glycoprotein spikes do, they are used to recognize and attach to the virus's host cell. Because viruses can only reproduce inside a host cell. They can't reproduce on their own. All right. So now if we move in a little bit farther, these glycoprotein spikes are attached, if you look right here, they are attached to a phospholipid bilayer. This is called the envelope. And the envelope is a membrane that is derived from the host cell's membrane. And we'll talk later about exactly how this works. All right, and this envelope, not every virus has it. If a virus doesn't have an envelope, oftentimes they do still have the glycoprotein spikes, but again, not always. All right, so let's move a little bit farther inside. This crystalline structure right here, this is our capsid, and that is the protein coat we referred to in our definition of a virus. All right, this capsid protects the contents inside, and the contents, these squigglies, that is the genetic material. And there's a reason I am saying genetic material and not DNA, because not every virus has DNA, all right? So just like eukaryotes and bacteria, many viruses do have double-stranded DNA. While others can have single-stranded DNA. They usually don't have more than one type, though. However, what makes some viruses unique is that they can have different combinations of DNA and RNA. So some viruses can have single-stranded DNA, which is unique to viruses. Or double-stranded RNA. All right. Now, if a virus enters a host cell and it only has RNA, that presents a problem because the host cell is only capable of making more making more DNA and copying DNA. A host cell can't copy RNA. Our cells don't have the machinery to do that. So, if a virus comes in with RNA, it has to somehow convert it to DNA. All right, and they do this conversion using an enzyme called reverse transcriptase. Remember that the process in a normal cell where DNA is converted to RNA is called transcription. If we go in the opposite direction, it is called reverse transcription. And with an enzyme, we always tag ACE on at the end. So reverse transcriptase is the enzyme that does reverse transcription. All right? And a virus that has reverse transcriptase and RNA is called a retrovirus. All right, so those are the basics of our virus anatomy and what we would find inside. Now, let's compare viruses and their cellular features, although they're not really cells, all right, their structures to bacteria and to eukaryotes. All right, so go ahead and draw this 3-part Venn diagram in your notes. Pause the video if you need to, okay? And we're going to compare viruses, bacteria, and eukaryotes. And we're going to start by taking a look at what all 3 of these have in common. So if we look at the structures of viruses, bacteria, and eukaryotes, all of them do have some kind of genetic material. Okay, whether that's RNA or DNA. All of them have membranes, whether that is the envelope in viruses, the capsule and the plasma membrane in bacteria, and the cell membrane in eukaryotes. They also can all have proteins in them. All right. Those proteins can take different forms, but they all have them. So now let's think about what is unique to viruses. Only viruses have capsids, and only viruses have glycoprotein spikes. Although other cells do have receptor proteins on their surfaces. Those glycoprotein spikes are fairly unique to viruses. Viruses are also the only place where we will find single-stranded DNA or double-stranded RNA. All right. Now let's think about some of those things that viruses do not have, but bacteria and eukaryotes do, right? So bacteria and eukaryotes both have ribosomes. And it is these ribosomes that are the key to why viruses can't reproduce on their own, right? Bacteria and eukaryotes use these ribosomes to do all of the functioning in their cells. Remember, proteins are the machinery. They are the transporters, the movers, the builders, the digesters, all right? Without proteins, you can't do anything in a cell. And remember that ribosomes make the proteins. So viruses do not have ribosomes, which means they can't make proteins, which means they can't carry out any cellular functions, right? They have to take over another host cell to do this. So that's the main reason why viruses need a host to reproduce. Bacteria and eukaryotes also have cytoplasm. They may have a cell wall, so I'll put that in parentheses. They may also have a flagellum. And both also have circular DNA somewhere. In bacteria, it's their main chromosome. In eukaryotes, we find circular DNA inside of mitochondria and inside of chloroplasts. Okay? So now let's think about what only bacteria have. Bacteria are the only ones that have plasmids, the small pieces of circular DNA. All right? They're the only ones that have a single circular chromosome. Eukaryotes, if they do have circular DNA, it will be inside their mitochondria and chloroplasts, and they will have many. Bacteria are also the only ones that have a double membrane on the outside. Remember, they have the plasma membrane as well as the capsule on the outside. All right, and then if we take a look at eukaryotes, eukaryotes are the only ones with a nucleus. They are the only ones with membrane-bound organelles. And then if we think about their chromosomes, all right, viruses, eukaryotes are usually the only one with many chromosomes inside instead of just one. Both eukaryotes and viruses may have linear genetic material as opposed to circular genetic material. All right, and you may think of some other things we need to add to this Venn diagram. If you do, go ahead and add those in and let me know so I can add them in the future. Right, but so that is our difference between viruses, bacteria, and eukaryotes. If we take a look at virus diversity, viruses do not all look the same. This first one, tobacco mosaic virus, was the first virus that was really discovered. And if you take a look, it has a capsid and some RNA, and that's it. It doesn't have anything else. All right. Adenovirus, which we had an outbreak of last year, has a capsid, some DNA inside, and you can see the glycoprotein spikes on the outside of it, used for attaching to and recognizing the host. The flu, our influenza virus, has an envelope with glycoprotein spikes, and it actually has multiple capsids inside it. And then this last one, our bacteriophage, right? Bacteriophages infect bacteria only. Their name actually means bacteria eater. All right, and so they have this strange alien landing pod formation. All right, so let's take a quick look at how viruses actually reproduce.
Scientists believe that influenza virus is spread from person to person through exposure to large respiratory droplets, direct contact, or airborne dispersal. Infection takes place mainly in the respiratory tract. Infection begins with attachment of virus proteins to a receptor on the surface of the host cell. The virus is then taken in the cell by receptor-mediated endocytosis. As the virus enters the cell, the virus is internalized in a membrane-bound capture vesicle that carries the viral core. The vesicle is transported on microtubules inside the cell by host proteins called kinesins. During transport, the membrane of the vesicle fuses with the membrane of the virus, and the capsid undergoes uncoating. The viral core, RNA, and proteins are then released into the cytoplasm, where they are guided by host proteins to the nucleus of the host cell. At the nuclear membrane, the viral core uses host protein channels to enter. Inside the nucleus, cell machinery is utilized by the virus to replicate the viral genome and make messenger RNA, mRNA. Some viral mRNA exits the nucleus to exploit cellular ribosomes to direct synthesis of viral proteins. Viral proteins go back to the nucleus to associate with viral RNA. These nucleoproteins again leave the nucleus and use cellular processes to travel to the cell surface. Viral surface proteins are made and processed in the cytoplasm and also travel to the cell surface, where they combine with the encapsulated nucleoproteins to form progeny viruses, which depart from the cell by budding. The virion now goes on to infect other cells.
Now is to do today's virtual lab or simulation. A copy of the lab has been posted to a bulletin for you on Verge. What you now need to do is do the virtual lab. Use your the internet to help you complete page 1, as well as our notes to help you complete page 1, and do this in your own words. The slides on Verge, which you can find in step 1 that you can see right here, will help you complete pages 2 to 3, and then think critically to complete pages 4 to 5. Once you have completed that virtual lab, you can go on to the next video.